Hi everyone, this is video 4 for the DNA replication, transcription, and translation video series. This is for the EOC review. This one's going to be very short, so I want to take a second to remind you of some things. Remember, this is not designed to be instruction only. This is designed to review. So if you are watching this is the first time you've seen any of this, this is not enough. You need to go back and find the relevant parts of this in the notes that I've given you through the year, and make sure that you're reviewing it. This is fast, and we've spent weeks on this kind of stuff in class. So I would encourage you to please go back and do a little bit more studying. So far we've talked about the idea of the structure of the DNA and how DNA copies itself during replication to ensure that new cells, after the cell cycle is over, new cells get their own copy of DNA. Then we talked about how DNA and RNA are very similar molecules, and some differences in RNA. We talked a little bit about what RNA might do, and I showed you 3 types of RNA. And today we're going to talk, in this video, we're going to talk mostly about messenger RNA, because this is transcription. Transcription only involves messenger RNA, and just so you know, that's going to be the little red molecule here. I've given you a drawing of it to kind of show you this. This is actually not just transcription, this is translation. And to some degree, this is also the DNA, or what we call the central dogma of genetics, and that is that information found on DNA is copied onto the RNA, and that RNA is taken outside, and that information is then in turn put into the structure of a protein, which is called a polypeptide, and then that polypeptide protein down here should be traits. So right now we're going to focus on this part, which is how we get the information from the DNA onto the RNA, and it's called transcription. The root word here is scribe, which means to copy. So trans means across, and so very simply we are copying across from the DNA information to the RNA information. A lot of times when you see the word transcribe, it's from one medium to another, such as I don't know if you've ever made a copy of a picture. If you ever copied it over to some other and printed it out, you turned it from one medium to another. A long time ago, we used to turn CDs into cassette tapes so that we could listen to them in the car. If you ever taken any CDs and turn them into an MP3, that's all transcribing, because it's kind of the same information but going from one mode to the other. Well, DNA is the same way. It's kind of the same information, but instead of having it on DNA, we're having it on RNA. And if you'll remember, this is important because the DNA is found in the nucleus, and it's like the master blueprint, and we don't want the master blueprint to be taken out of the nucleus where it could get damaged. So instead of taking the master blueprint out, we just take and make a copy of the parts that we need from the blueprint, and we take that copy out. And if something happens to that copy, no big deal, we can make another one, but at least the original copy wasn't damaged. And that process of making that copy is called transcription. Now, this happens both in prokaryotes and eukaryotes. Remember, prokaryotes are pretty simple. They just got kind of a circular shaped DNA called a plasmid, and they don't really have to do a lot. It's all outside of the nucleus, and it just happens. But for eukaryotes, because the DNA is so large, and it's found inside of the nucleus and protected, we have to go through this several step process. This process kind of happens in prokaryotes, but it's not that complex. And they use the RNA, but it doesn't have to travel out of the nucleus because prokaryotes don't have a nucleus. But eukaryotes do, and so this process involves making the copy and then moving it out of the nucleus. And that's what we're going to be talking about now, is making this copy of the gene off of the DNA and onto the RNA, messenger RNA molecule, and moving that molecule out of the nucleus. It looks a lot like replication, because that's how the DNA works, is it can be open and closed. And so to start out with, we have the RNA polymerase finds where this starts, and it moves in and it unzips the DNA, and then we start having, along comes a different type of enzyme, which will start making a copy and adding the nucleotides to this RNA strand, and then it closes up and it's done. Now, there are 2 strands for the DNA, as you know, and technically we're making a copy of this side. Okay, and I want you, if you think about it, that makes sense, because this strand should look just like this side when it's done, except that, instead of thymine we have uracil, but other than that it should be the exact same code. So the gene that we're copying is actually over here on this side. It's called the coding strand. This side is called the template strand, and that's what we make a copy from, is the template strand. Okay, but the actual information, the gene, is over here. So the RNA ends up being a copy of the coding strand, not the template strand. And I'm just letting you know that sounds like a test question. As it comes loose, in fact, take a look here. Here's the template strand, and here's the coding strand. And if you took out this for a few minutes and just noticed A bonds with T, T bonds with A, G bonds with C, A bonds with T, all the way down. So when we go to make the RNA strand, we're actually going to take a complement to this side, AUG, AUC, and that actually looks just like this side, except we replace all the T's with U's. So we're technically copying the coding strand side, even though we're attaching it to the template strand side. Okay, but it's a pretty straightforward thing. This polymerase opens it up, and then along comes another enzyme. Honestly, I'd tell you what it is, but I forgot. And it starts adding those nucleotides as it goes, and then once it gets to the end of the gene, the RNA polymerase stops, the DNA zips back up, and the RNA is moved out and it's done. I want to show you another picture of that. I don't know why that did that. Okay, but I want to show you that again because there's the idea of these things called promoters, and promoters tell us when to start and stop. This transcribed region is actually the gene. Okay, well, it's all the gene, but this is the gene. But the polymerase, because remember, this thing is huge. This DNA strand is 3 billion pairs long, and we only want about 1,000. So how do we know where to start and to stop? And there's the polymerase knows to look for these promoter sites, and it goes to these promoter sites, which is a set of 3 pairs. It goes right here, and it says, "Oh, that's where we start," and then it starts moving along the DNA until it gets to another promoter site that says "stop," and that's how we know where the gene is on the DNA strand. RNA polymerase is not smart. There's no intelligence involved here. It's just the mechanism is set up. It's like your car works. It doesn't have much of a brain. It just does what it's supposed to because of the way that it's built. And the same thing here, because of the shape and the way the DNA works, we can have this happen. And so we move here and we copy a very short part of the DNA. It's called a gene. And once that gene gets copied over into the RNA strand, we zip it back up and we're done, and now we have our working copy, and that's the end of transcription. Okay, not much more to tell you here. It's that straightforward and that simple. The I know that you see this 3 and this 5 prime. I've kind of explained that before, but again, at this level of science, I don't think we need to understand that specifically. Remember, they are anti-parallel, meaning that one goes one way and one goes the other, and that's what the 3 and the 5 prime mean. It really has no effect on opening and closing this thing. It unzips, the RNA is copied, it zips back up, that's that. So that's going to end this particular video. Gosh, it was only 8 minutes long. I can't believe that. So go ahead and take the check yourself quiz on this just to make sure you understand it. If not, watch the video one more time. I don't want you to make this any more complicated than it is. Just really 2 things to know. We unzip it, make a copy and zip it back up. And how do we know where to start and stop? We have these recognition sites called promoters. Okay, that is the end of video 4. I will see you at video 5. Go take that quiz.